Good morning to one and all. Welcome to the second day of the webinar in the series of Faculty Development Program titled Plant Protection, Current and Future Perspective. I would like to welcome to our today's first speaker, Dr. Biswajit Patra, who is working as an assistant professor in the Department of Agriculture and Entomology, Uttar Banka Krishik Vishwavidyalay, Kundibari, Kuch Bihar, West Bengal. He is going to present on the insecticide resistant mechanism. Sir, good morning, sir. Now you can start your presentation by sharing your screen, sir. Good morning, sir. Biswajit Patra, sir. Am I audible? Sorry for the delay. We're facing some internet issues.
Hello, Biswajit Patra, sir. Hello. Biswajit Patra, sir. Hello, Biswajit Patra, sir. Sir, please unmute your mic, sir.
Just one thing. Hello.
Yes, sir. Yes, sir.
Just a minute. I am just starting.
Yes, sir. You.
Am I audible to all of you?
Yes, sir.
Yes, sir. You are audible. You can start.
Start sharing your screen, sir.
Just I'm sharing my slide. Just a minute. Sharing to. Nihal, just a minute. Just wait. Just wait.
Okay, sir.
Mhm. Good morning, all. Uh, there is some problem in Biswajit Patra sir's network. So I would like to request Dr. Abhijit Ghatak sir to start their presentation. First of all, I welcome Dr. Abhijit Ghatak sir. He is an assistant professor in Department of Plant Pathology, Bihar Agricultural University, Sabor, Bihar. Sir, I welcome on behalf of our university, that is Centurion University of Technology and Management, Odisha. Sir, you can please start your talk. Now, mic is up to you, sir.
Okay, thank you. Thank you so much, Ritesh. Let me first share my screen.
Yes, sir.
So I think it is visible to you.
Yes, sir. It's visible, clearly visible, sir.
Okay. Thank you so much for having me on this nice platform to interact with you people and particularly with the students and the faculty of this institute. I therefore thank to the organizers and the dean, particularly of this university, for inviting me for having to share my experience in this field with you people. And it will be definitely I think knowledge exchange platform so that I can share with new avenue, new vista of this plant pathology. So this will be nice. At first, this is my brief introduction in the slide. Myself, Abhijit Ghatak, who is working in Bihar Agriculture University since 2012. It is almost 8-year experience from my side. My education, as far as my education is concerned, so I did my PhD from GPPAT Pantnagar, and also my research work was conducted in Erie, Los Angeles, Philippines. Before that, I was in BHU Varanasi, where I earned my MSc in mycology and plant pathology. Some of my professional experience is also briefed here. I played a role as a research associate in Almora of Uttarakhand, that is Northwest Himalayan Hills. And thereafter, I also did a senior research fellow in GPPAT Pantnagar in a DBT funded project. And currently, I'm having an international project in RIA from France. And before that, I had also conducted some experiments of cements from Mexico. So this is my brief introduction. I think we are having in this program, we are having some students also from UG background particularly. Please let me clear so that.
Yes, sir. Some UG students, some MSc and PhD also are in this webinar, sir.
The MSc and PhD also.
Yes, sir. Along with our faculty members.
Yes. Okay, sure. With my topic, that is fungal root-borne disease management of pulses using post-mushroom substrate. So in this topic, entering into this topic, I would like to share a review paper published in Plant Disease in way back in 2011. I think yes, 2011 in Plant Disease. That is by American Phytopathological Society, whereby they claimed that a new disease. Actually, the disease is old, but it is new to this current era due to the elevated environmental temperature as a resultant of climate change. So therefore, this problem is arising from acute to as emerging problem due to the warmer condition of our environment. So this is chickpea dry root rot. They actually claim that it is very widespread disease across. In fact, infect across several crops, including sorghum, many pulses, and other vegetables also. So this is caused by Rhizoctonia bataticula, or you can say this is Macropomena facilina. So these are a few symptoms of this disease in the field as well as in close view. In the field, you can see the space where this infection was happened and the plants are used to be died, collapse, actually total collapse. It is, you can say, it is look like a wilt symptom from the outer side, but it will be your experience when you uproot the plant. When you uproot the plant of a wilt infected plant, so you need to pull it out with implementing a minute force with your hand. But when you are uprooting such kind of plants infected with Macropomena facilina for this dry root rot or charcoal rot disease, so this will pull out very easily with very limited force because of this integration of these secondary roots. See, there are no secondary roots are available per se in this figure, and here some are many secondary roots are vanished even in the middle this sample. So this is a very important symptom, and it will come. It will you will know about this disease when you have experience for pulling out such plants. So these are some survey, several fields in Bihar. It is near in Jamalpur district, Jamalpur in Munger district, and this is Mokama Tal in Patna district. These figures. See, there are several patches in the field where these infected plants are almost vanished, and some are also visible, which will be going to be vanished. This is the Tal area of Patna district. This is very unique agroecology where this is a saucer-shaped ecology, agroecology actually, whereby in the central portion of this ecology, flood used to be covered around the year, but for 4 to 5 months, the farmer used to sow pulses, particularly lentil, and 80% lentil and 10 to 15% chickpea, and at some patches and some spots, some mustard you can see. So this is very means they used to claim this is a ball of pulses in Bihar. So in this region also, this pathogen is in very degraded condition, and you see the severity of this disease. It is approximately, I would say, in this field, it was approximately 70% damage was caused at that time. The figure was taken in January 2019. So looking into this aspect, this for management of this disease with some biological means and implementing some amendment, that is amendment into the soil without implementing this chemical strategy. So we look forward for mushroom waste, that is post-mushroom substrate. So this is nothing. This is actually the substrate that is left after the mushroom harvest. When you pluck the mushroom crop, after that you can use this bag, this post-mushroom waste as in this experiment. I will show you this experiment. So this post-mushroom compost is also known as spent mushroom compost, and as it is soil-like material which represents the compost substrate remaining after harvesting mushroom bodies, fruit bodies. This substrate with innumerable mushroom threads, as you people are knowing probably from plant pathology discipline, collectively referred to as mycelia, is biochemically modified by the mushroom enzymes into a simpler or more readily digestible components. Ultimately, This is the mechanism actually. Mushroom mycelia can produce a group of complex extracellular enzymes which can degrade and utilize the lignocellulosic waste in order to reduce pollution. And therefore, it has been revealed that recently that mushroom mycelia can play a significant role in the restoration of damaged environment. What it called? It is called myco restoration through means mycus, that is mushroom. So post mushroom substrate as myco restoration. Myco restoration is the use of fungi to restore degraded environment. Some mushroom can be used to remove toxin and heavy metal contamination from ground are called myco restoration. Myco restoration can be performed into different ways. There are 4-tier systems: mycofiltration, mycoforestry, mycoremediation, and mycopesticides. So our study is mainly focused on mycopesticides. I will come up into my further slides. So mycopesticides, we will use this strategy for plant disease management. So there are several works have been done previously using this post-mushroom substrate in Fusarium, Rhizoctonium, Macrocomia, and several other fungal plant diseases. But it was never standardized in under field conditions. It was simply some greenhouse experiments and other. Hereby, I will show you 4 years of field experiments and then OFT, that is on farm trials in farmers' fields, and thereafter we have standardized our results for 2 years again. So it is 6 years actually. So this is a compact output of our experiment. This will be. So these are okay. I'm going further. So coming into my main program, this is the technical program actually. That is field and laboratory experiment. Will cover then laboratory experiment, temperature kinetics in the treated experiment plots, and population count, that is CFU. Many of the students, particularly from UG background and newcomers in MSc discipline, may not be knowing about CFU, that is colony forming unit. This is the way how we count a population of a fungus in a given sample. So it will be dealt in further slides. Chemical analysis of mushroom waste and experimental plots also done, and thereafter finally assessment of disease development, and finally the yield assessment. So these are the gifts of mushroom bags. What we are saying here as post mushroom substrate. See here, the original mushroom have been plugged, and this is left aside in a roadside. So this will invite also pollution and invite flies and other polluting agents, which will which used to be produce foul smell in the area and causes for different kind of diseases like malaria and cholera, etc. So these are the source of pollutant. And beyond this, some plastic bags are actually it is covered with. It used to be covered with plastic bags, and these plastic bags are also as a pollutant, which causes damage to our environment. So these were 2 major objectives of my program. That is determination of the potentiality of mushroom waste on survival of Macrocomia facilina, and the other one is assessment of the impact of mushroom waste, that is actually post mushroom substrate on chickpea. I worked on chickpea here and partly on that is field pea and lentil. So on charcoal rot disease, I'm here showing you in detail for charcoal rot disease, but I also taken the assessment for wilt, that is Fusarium wilt, and for Sclerotium rug or southern blight, that is Sclerotium roxai. So 3 major diseases were covered, but here I have, due to lack of time and slides actually, so I took here for charcoal rot disease because this is new to everyone. So the methodology we covered for objective 1 was chemical analysis of mushroom waste and experimental plots, and thereafter the population, we counted the population of the fungus in the rhizospheric zone. And in the second objective, we went for assessment of disease development, and finally yield estimation was done. Before entering into our main program, I would like to let you know about Trichoderma, what we used here in our study. So it was actually locally isolated Trichoderma, from a hot spring, and it can tolerate up to 45 degrees centigrade. And see some plates from particularly UG students and newcomers in MSc students, that you can see this is Trichoderma, our Trichoderma, and this is a pathogen. It is actually Fusarium. So it can it is inhibiting the progress of Fusarium, and ultimately it will cover up the slide, the whole plate actually. So the main feature of this Trichoderma is it can sustain up to 45 degree temperature under laboratory condition, and it was therefore taken for our program into the field because due to high or elevated temperature in the current situation, due to high, elevated temperature, this pathogen Macrophomina facilina is making nuisance to the crop, to several crops, particularly in pulses. And it was again confirmed with 18S ribosomal DNA amplification. This is our isolate, that is TVB1, and several other Trichoderma were also taken into account for understanding their length. So our Trichoderma isolate, that is TVB1, came in between approximately in the length of its nucleotide is actually 600 base pair. And it was further confirmed with sequencing for ITS region, that is intrastructural spacer region, for our isolate, and it has been it is shown 99% similarity with Trichoderma asperellum. So therefore we confirm that our isolate is Trichoderma asperellum. Further, this Trichoderma was actually mass multiplied on fly ash. Fly ash actually the output from charcoal or coal actually. It used to come out from thermal power stations. We taken it from Kahalgaon in Bhagalpur district, where an NTPC is established. So here, this is the procedure how we produce this Trichoderma into mass multiplied fly ash based formulation. So different studies were made previously. Finally, we concluded for fly ash, and it was actually released from our university in the name with the name Trichoderma 1. And finally, I would like to show you this slide of our isolates of the pathogen, that is Macrophomina facilina. See, this is Macrophomina facilina isolate, and see their variation. Some are very fluffy, some are sparse, some are very compact colonies. For example, these are some compact colonies, and these are some sparse colonies. And see, they are very fluffy in nature. And some see the variation in color as well. Some are grayish in color, some are whitish in color. Okay, so there are several many variation in among these isolates of our Macrophomina facilina pathogen. And this is again some symptoms of chickpea roots infected with Macrophomina facilina. Even after harvest, you can see after harvest, these are after harvest samples, where you can see this blackish charcoal covering on the root surface, over the root surface. And these are actually came very late in the end of the season, and it contributed for yield reduction. Some other diseases for the students actually, I would like to show you some symptoms. For example, this is Sclerotium rossi, and here you can see the roots of Sclerotium rossi infected roots of chickpea plants, where you can see this look like a polio person, where you can see in the collar region actually the infection is happened, and thereafter the entire root from the beneath of the infection, the entire root used to be very shrunken and look like polio-like symptoms. And these are some isolate variation of isolate producing sclerotia. See, there are 1, 2, 3, 4, 5 isolates were there. I'm just showing for your understanding, but they are varied in sclerotia production. Sclerotia are the resting structure of the pathogen, look like a mustard seed, smaller than mustard seed, in fact. So, and these were mass multiplied while. Yes. If anybody,
please mute to all. Ritesh Kumar, there are some background sound. Please mute to all.
Yes, sir. Yes, sir. You
Borrani ma'am, please mute yourself.
Sir, you please continue.
Okay. Otherwise, you can Ganesh may please mute the other. So these are some examples of Fusarium of this forum. This is please.
Hello.
Sir, please continue, sir. Please continue.
Abhijit, sir, please unmute yourself, sir. Yeah, unmute.
Yes.
Yes, sir. Yes, sir. Please continue, sir.
Okay. Thank you. Yes. So these are some symptoms of Fusarium oxysporum. That is, here you can see starting. It is an infected lentil plant, and it is started to wilt. And there are several very smaller seedlings, 3 weeks or 4 weeks old seedlings are there. And there is some variation among the isolates, whereby an isolate is producing pinkish color of pigment in the medium. And these are again for mass multiplication, mass multiplied sorghum seeds, which are used to be inoculated in the field. And just wait. Yes. So this is our in the field program. Before entering into the field, we calculated the values of our mushroom post mushroom substrate. What are their combination of in different parameters? For example, carbon, nitrogen, phosphorus, and potassium were estimated. And here you can see different in different conditions. We plotted our field in under different treatments. The treatments, we will show you the treatments. And this is actually sorghum seeds dipped into in water for overnight, and this is mass multiplied our pathogen Macropomena facilina, which were again scattered on the experimental field. These are the actually experimental treatments. So here MW means mushroom waste, and you can correlate this with PMS, that is post mushroom substrate. This is the same. So we have taken 3 different volume of mushroom waste, that is 6 ton, 8 ton, and 10 ton per hectare. individually and along with trichoderma, that is trichoderma with seed treatment. Please mind, trichoderma is used to take 4 to 5 gram for lentil and 8 to 10 gram for chickpea per kg seed. But here we used only 2 grams per kg seed. So we have reduced the amount also, because we are giving them the semi-decomposed biological matter, that is mushroom waste or PMS. So beyond this, we have taken also FYM, that is farmyard manure, and vermicompost. And we have kept a positive control, that is carbendazim, acid treatment, and a negative control where nothing were applied in the check. So this is a field image in 2019. This was an experiment in 2017, which were failed actually. The first experiment was failed because in several plots germination was a problem. No germination were there. So therefore, so big ACM, that is standard error were generated, standard error of mean, and thereafter 2018 and 19 and onward 2020 were quite nice. So these are the results of 2018, 19, and 2020 year for 3 years. And 2021 I have not included yet for this year. So we have 4-year actually experiment. So 2021 were not included in this study. So in 2018, so it was insignificant among the treatments. So there were no variation actually. So it was non-significant. But in 2019 and 2020, there were some variation we got because of high amount of disease. Because at this time, the winter shower were totally absent in these 2 years in our location. So due to this winter shower was absent, and thereafter high disease were occurred. It was approximately 16%, and in this year it is over 13%. So thereafter we have got very nice trend of our treatments, where you can see that is MW, that is mushroom waste 6 ton per hectare, and trichoderma acid treatment, when we are giving this, providing this to our plots, to the field. So it has given very comfortable result in this tree. See, they are insignificant among these three. So we can take this smaller amount of source of mushroom waste. So mushroom waste with 6 ton and along with trichoderma has given good result, the minimum result. However, carbendazim were provided very as far as yield is concerned, this is the result of our disease. As far as yield is concerned, carbendazim has provided greater yield. I will come back. And it can be correlated with soil temperature kinetic. It has taken for each year, but for the first year, that is in 2018, it was taken only 3 times. Later on, we taken for 5 times. So see, the date is mentioned, that is middle of February, 13 February, then early 1st March, and then middle of March. So you can see here that in the middle of March, it is like concave in shape when the temperature is increasing, the day temperature is increasing. Here you can see the timing of temperature, that is 7 a.m., 10 a.m., 1 p.m. and 3 p.m. Okay, and it is temperature. Okay, and but in the middle of March, around Holi time. So in the middle of March, the minimum soil temperature is used to be varied approximately 5 degrees centigrade. See here, it is 26.5 and it is 32. The mean day temperature. It is varied approximately 5 degrees centigrade. It is 25 and it is 30, because it is 10 centimeter depth. These all are 10 centimeter depth, and these 3 are 5 centimeter depth. So in 10 centimeter depth, the temperature is much more cooler. So it is 25.2 degrees centigrade and this is 30.7 degrees centigrade. Means approximately 5-degree variation is there in this 15 days. And here is the time when this disease used to be shoot up, risen up. Because it is thermophilic pathogen, it loves temperature. And therefore, this pathogen is very causing very dreaded problem in such conditions. And see, for next year, 2019, for next year, we have taken it for 5 times with the same timing, that is 7 a.m., 10 a.m., 1 p.m., and 3 p.m. For one assessment, for one-time assessment, it took approximately 1 hour, means 7 to 8 a.m. Actually, it is 10 to 11 a.m. For entire experiments for each field, it took approximately 1 hour time. So this is again, you see, there is no variation, no much more variation. 22.1, 22.2, 21.3, and this is 21.5. So there is no much more variation in between approximately 15 days. This is 3rd of February, this is 15th of February, approximately, okay? But here, this is very slight change, slight increasing change. But in the middle of March, again, you can see it is increasing approximately 4 to 5 degrees centigrade. Okay, so in this time, this pathogen is making problem. So furthermore, we had counted the population of Macropomnia fasciculata from our rhizospheric zone in different plots, where you can see this is CFU. For newcomers, I would like to say that they are the colony forming unit. Actually, in one unit, how many colonies are formed? It is determined with this formula and calculation, and thereafter we calculated the CFU on different time intervals, on different observations, like first observation, second, third, fourth, fifth, sixth observation. Okay, so here you can see similar trend at each and every condition, each and every year, similar trend we have found that there is very high CFU were obtained in Czech, that is controlled, and thereafter when mushroom waste is alone supplied. And if you are supplying Trichoderma along with mushroom waste, it will be reduced. See in each and every year. And you can see the final observation, that is 6th observation for each year. This is the final observation. You can see again, when you are providing Trichoderma along with mushroom waste or PMS, so it will be reduced. The colony forming unit will be reduced. So lower population will be there in the rhizospheric zone. Otherwise, this pathogen is increasing their actually population without the presence of Trichoderma. And in case of carbon dioxide, it is best at each time. So this is assessment of yield. I'm not going to let you know what yield we obtained. I am just showing you that percent change of yield from check. If you are, suppose your check is 0, so how much percent change you will get your yield in comparison to check. If your check is 0, you are getting 0 yield from your check, so you will get 35.6% more higher yield than your check. So if you are getting, for example, you are getting 1000 kg per hectare from check, so you will be getting 1350 grams yield from this treatment. So these three are insignificant. These three are insignificant among them. So we can again see, we can choose this one because of lower supply of PMS. So here you can see the benefit cost ratio. Again, we have calculated the benefit cost ratio, whereby you again see the best were found for PMS 6 plus Trichoderma. Actually, these three are insignificant, but we can choose this one, lower one. So this was calculated with the net profit. So for example, if you're investing 1 rupee for check or control, you will get 1.93. This is 1.93 in return. So if you are investing 1 rupee here, so you will get 2.24 rupee out of this treatment. This were actually our 3-year experiment. This year I did not calculate. This year experiment I need to calculate. Actually, these are the results of 3 years combinedly. And later on, actually 2018 and onward, we also conducted on different pulses with limited treatment setup. So we finalized with Trichoderma and PMS along with positive control and negative control. Trichoderma and PMS along with positive control and negative control in each condition. For example, in chickpea, in lentil, or in pea. In field pea, we took 2 varieties, that is Azad Matar and Punjab 89, which is very promising variety in our North Indian plain zone. So we calculated for 3 different diseases. 3 diseases in total. You can see here. Finally, total root-borne diseases, about root-borne diseases. In case of chickpea, you are getting 24.6% of problem in control. However, when you are using carbendazim, this problem is reduced down, and it went for 6.7%. And in between, when you are using PMS along with Trichoderma, you are getting only 13%. And please mind, this PMS is only 3 tons. Previously, we were showing it's 6 tons per hectare, and here we are using 3 tons. We again reduced down for checking to understand whether this will work or not. So this is 3 tons per hectare at every case, 3 tons per hectare. So you can see the similar trend for everywhere. We are getting nice disease there at those years in 2018, 19, and 1920. This is 1920. The similar trend we obtained. Okay, because of very less or no winter shower in those years at our location. So we got very good results, particularly for dry root rot. And these 2 diseases are very common at our place. So this is in our farm field, and this is the result of our yield, that is presented in terms of quintal per hectare for chickpea and lentil, and this is for ton per hectare for garden pea, field pea. So at every case, the trend is similar for this 2 years. Again, in case of we combined those 2 years. I showed you this for individual years. We combined in this table. We combined everything for 2 years. In the mean, actually, you can see the similar trend is there. It is 23.5%, and you are getting approximately 14% injuries when you are using PMS along with Trichoderma, and when you are using carbendazim only, so it will be around over 7%. Problem is there. So as far as yield is concerned, again you can see the highest yield was obtained from carbendazim applied plots, and in between the PMS and Trichoderma were given in between result with 14.84 quintal per hectare, and this control plots were given 12.37 quintal per hectare. Similar trend for each and every crop is there. Okay, so it was calculated. It is benefit cost ratio for the same for the 2 years. You can see if you are investing 1 rupee, so in control you will obtain 3 rupees. Okay, so if you are investing 1 rupee here, you will get 3.5 rupees. And if you're using 1 rupee for carbendazim, so we will got 4.16 rupee. Okay, so carbendazim is giving a very nice result at every time for every situation. So this is okay, but this is chemical management strategy. We are looking for the organic management strategy, and also using this PMS because of making problem to our environment, and it will supply nutrition to our field, and it will encourage the soil parameters, other soil parameters to helpful it will helpful to our soil to increase its value in future. So you will see if you are investing 1 rupee here, so in control you will get 3.2 rupee. So the same trend is obtained from these two combined year. This is benefit cost ratio, and we plotted the such experiment in for OFT in Buxar, that is West Bihar, and in Arwal, this is in Central Bihar. Okay, the same experiment were given to them for OFT, that is on-farm trial, OFT. So again, we have got here, we increased our treatment were also given, that is PMS 6 ton per hectare and PMS 3 ton per hectare for each cases. So in Buxar, it was conducted with chickpea, and at Arwal, that is in Central Bihar, it was conducted with lentil. So again, you can get the nice result, nice trend, but it is nearly insignificant between the seed treatment in this year. It was insignificant between seed treatment, that is with carbendazim and with PMS6. But when you are using PMS3, so the disease is higher than PMS6, and it is in control. We have got very nice disease pressure. The same thing is there for lentil also, but in lentil there is variation in seed treatment and in PMS6. So this were conducted on the farmer's field, and here you can see the yield at Buxar for chickpea and yield at Arwal for lentil. The similar kind of trends were there for these two cases. This is actually percent change to control. If you are having only 0 from your control plot, so it will provide you 18.2% more in PMS6 plus Trichoderma, PMS3 plus Trichoderma, and 2% more you will get when you are applying PMS6 along with Trichoderma. So at both of the cases, carbendazim has yielded the greater result, for the yield. So it is my conclusion of our study. As far as disease incidence is concerned, it was ranged between 1.3 to 4.7 during 2017-18. It was very less disease pressure were there, but in the coming years, 2018-19 and 2019-20, we have got up to 15% and up to 12.5% disease pressure in these 2 years. This year value I did not calculate, again this might. So minimum temperature indicated for increasing the possibilities of charcoal rot disease development. Minimum soil temperature, what I showed you here in this slide. Here, minimum soil temperature. This is helpful for providing a good support to this disease, charcoal rot disease. So minimum temperature indicated for increasing the possibilities of charcoal rot disease development. The plots applied with PMS6 plus Trichoderma rendered 35.6% greater yield compared to the control plots. For the application, it is well defined at every slide. Application of carbendazim rendered 56.5% greater yield than control. This was the result of our experimental plots, not in farmer field. The other experiments and OFT, that is on-farm trial, OFT suggested to use 3 tons PMS post-mushroom substrate per hectare plus trichoderma. Please mind here, we have supplied only 2 grams per kg feed for managing major fungal root-borne diseases in pulses. Across several pulses, we conducted our experiments, including chickpea, lentil, and field feed. And overall, the experiment indicates for a suppression of chickpea charcoal rot with post-mushroom substrate. This is an agri-based amendment. This is possibly due to this need to be unveiled in future research. This is possibly due to enhancing rhizospheric activity of beneficial microorganisms, including Trichoderma. Means our PMS is providing support as food material to Trichoderma and other beneficial microorganisms, other beneficial bioagents to flourish themselves. And thereafter, they used to kill the pathogenic organisms and sustain themselves in the root zone. So these are the overview of our research. This is the key outcome. This technology, I'm going to propose this actually in our council in future. So this technology has produced lower root-borne disease occurrence and greater yield have been obtained from PMS plots, that is post-mushroom substrate plus trichoderma as seed treatment. So specification and salient technical features. These 2 technical features were there. That is application of PMS and seed treatment with trichoderma rendered lower yield, lower disease under field conditions compared to control. And as far as yield is concerned, greater yield greater quantity of yield harvested from PMS and seed treatment with trichoderma treated plots. So this is take-home message. You can go with PMS 3 tons per hectare and seed treatment along with trichoderma, that is 2 grams per hectare against various fungal root-borne diseases of pulses. So our coverage area, this is very wide coverage area of this research is coming out. That is, this technology is also viable for field of upland conditions and moisture conditions, because in upland or moisture conditions, Phomina fasciculata, they used to perish. Kindly mute. Some disturbances. Okay. So this technology is also useful in the late sowing cultivation system, because in when you are harvesting rice in late, for example, in late of November, so pulses sowing is going used to go for up to first week of December. And in fact, up to 10th of December, the farmers is to sow pulses of chickpea or lentil. So in late sowing system, this is a major problem. That is Macrophomina fasciculata causing charcoal rot or dry root rot is the major problem in late sowing system. So this will be helpful to manage those diseases, such diseases which is thermophilic in nature. So in view of climate change, so this technology could be adopted as the provided agri-waste ensure water holding capacity for a longer period. Again, this is one benefit. Also, the technology can be implemented where organic production system is targeted. Simply, we are targeting to manage our diseases using organic inputs. So, impact on the income of the farmers. So, it will save the loss using agri-waste of post-mushroom substrate and seed treatment with trichoderma. This will ensure farmers' profit. It will also help in promotion of pulses cultivation among the farmers, and it will assist in the reduction of environmental pollution around the season. So this is my small team in our research plots. So these are very nice plots actually, as far as one plant pathologist is concerned. We are developing here 6 plots, and these were actually the damaged plants that were uprooted, counted and uprooted. and if you use this practice is going up timely, and so these were damaged by the infection of our target passages, concerned passages. So thank you so much.
Uh, thank you so much, sir. In this arena of global warming and climate change, which attract a different type of pest and disease in last couple of years, and farmers are using different types of chemicals, which are not only increasing their income but also affect our food quality. So these type of approaches or these type of experiments would definitely helpful for the farmers and all the agrarian communities. Once again, thank you very much, sir, for your innovative talk. Now, I would request our faculty members, if they have any queries, they can ask. I would request our participants, if they have any queries regarding this, they can ask, please. Okay, I would request Dipan sir to please read the queries from the chat box.
Hello. Hello, sir. Am I audible, sir?
Yes, yes, yes.
Yes, sir.
Yes, sir, you are audible.
Yes, sir. Thank you, sir. Thank you very much for your informative talk. We learned so many things from you. Thank you, sir. Sir, one student of MSc AG from BCKB, he has some queries, sir. He has asked in the chat box that how are the conidia of Trichoderma can be harvested from the stock culture. It was his question, sir. He is having the
What's
queries
the conidia of Trichoderma?
from the stock culture.
Yes, actually, we used to keep this, take this plug, mycelial plug from our PDA, and we used to inoculate for in the substrate where it is used to be mass multiplied, and thereafter, after several process, several steps are there. Along with me, one more scientist is working, who is taking care of this trichoderma section. So thereafter, we used to inoculate in mass multiply in the fly ash. So there are several steps are there, many technical steps.
Okay, sir.
पात्रा सर नहीं है मीटिंग में.
Thank you, sir.
Excuse me, sir.
Yes,
Am I audible, sir?
yes.
Sir, actually, what happened in Assam Agricultural University, we are using for mass multiplication of trichoderma, talcum
Your-
powder we are using.
Excuse me.
What
Your,
criteria using for this one talcum powder for mass multiplication?
can you please repeat yourself because your voice very interrupting?
For mass multiplication of trichoderma, we are using talcum powder. Hello? Am I audible, sir?
Yes, yes.
Yeah, we are using the talcum powder for mass multiplication of trichoderma. We are generally practicing, we are making the biopesticide, trichoderma viridie-based biopesticide using the talcum powder.
Right.
Whether you have applied this or not, sir?
No, we did not use talcum powder. We used this because this is widely available. We use.
Sir, excuse me, sir. Sir, kindly mute these 182 Psych and Munina. There are some noises coming, sir.
Thank you.
Mostly welcome, sir. Now they are muted, sir. Now we can start.
Dipan sir, any other queries?
Sir, from the chat box, we got this much, sir. From participants, those who are present, including students, they can ask if any queries there, our students or participants directly through this Zoom meeting.
Yes, yes.
Okay, sir. If anyone has no query, then we'll move to another talk. Once again, thank you so much, sir, for your innovative talk. This will definitely help us all. Thank you so much, sir. Thank you so much.
Okay. Thank you. Thank you for having me in this platform. Thank you so much.
Thank you,
All
sir.
the
Okay. Good afternoon, everyone. I'm very grateful to introduce our second speaker, Dr. Biswajit Patra, Assistant Professor of Agriculture and Entomology, Uttar Banga Krishi Vishwavidyalaya, Kuch Bihar. Sir is going to present on insecticide resistance mechanism. Sir, over to you, sir.
Yes.
Please share your
Thank you.
slides.
Thank you, Dr. Nihal. Thank you to Dr.
Thank you.
Abhijit Ghatak also for his nice presentation. Is it audible to everyone?
Yes,
Am
sir.
I
It is
audible?
audible, sir. Yes, sir. You are audible, sir.
Okay. So should I start my screen sharing?
Yes, sir. Sure, sir.
Okay. Just a minute.
Abhijit, sir, please unshare your screen, sir.
Yes, it is taking time due to network problem most probably. Is it fine?
The slide is visible.
Okay, should I start?
Yes, sir. Please, sir.
Today I am going to present on a topic that insecticide resistance mechanism and management. Myself, Biswajit Patra, Assistant Professor in Regional Research Station in Field Zone, UVKV Kalimpong. At the outset, I would like to show you a picture or image. I hope all of you can recognize this very famous person.
Sorry to interrupt you, sir. Please maximize your screens.
Okay. Is it fine?
Yes, sir. Please continue.
Okay. Now, please. The next image is this one. The first image and the second image, both are interlinked. The first one is the picture of Dr. A.L. Millender, and the second one is the image of San Jose scale, Quadrus pediotus parsiniosus. So he is the person who first reported insecticide resistance in the world in the paper of Journal of Economic Entomology in the year 1914. We can see the paper. The title of that particular topic was, "Can insect become resistant to sprays?" That particular answer was the yes, insect can become resistant to sprays, and that particular paper was published in the 1914. From that 1914 to till date, almost within these 107 years, numerous pests has developed multiple resistance against the available insecticides. A quick visit to the Arthropod pesticide resistance database will make it clear that how many pests have developed resistance in today's context. So resistance, be it insecticide resistance or acaricide resistance, whatever it is, no doubt it is one of the serious problems to mankind. Insecticide resistance remains one of the greatest challenges in the modern agriculture and public health pest management. And it is crucial that it is tackled effectively. Indeed, resistance is everyone's problem. So, due to the resistance problem, farmers they tends to use more and more application of the pesticides, which ultimately leads to environmental pollution and toxic residue in the food. This is the problem of the resistance. So, not only the insect, see the Helicoverpa, Plutella, Spodoptera, Bemisia, Myjas persicii, Empoasca, Nila parvatalugens, almost the list is endless. Almost every insect has developed some percent of the resistance or some degree of the resistance against the available insecticides. See the mite, Heteronicus artici, see the mites, Oligonicus coffee, see even the storage pest has developed some degrees of resistance against the available insecticides. Tribolium, even the household pest, Musca domestica, even the public health importance pest like mosquito, Aedes mosquito, Culex mosquito, whatever, even the veterinary pest has developed the resistance to the available insecticides. See the graph. If we see the graph, we can easily understand that the resistant species is increasing day by day. Even the compounds against which the development of resistance is reported from different parts of the corner, it is also increasing. Almost the species that they are developing very quickly. In fact, the species of different insect or mite or whatever it is, they are developing the resistance against the available insecticides very quickly. See the insect resistance in India. First instances of resistance was reported from mosquito, that is Culex fatigans, against the DDT in the year 1952. Then it was reported in bedbug in Cimex species. It was also against DDT in the year 1953. As far as the veterinary pest is concerned, the cattle tick, Bufila species, also developed resistance against in Linden, and it was reported in the year 1963. See, as far as the agricultural pest is concerned, all of you know that Singhara beetle or Garrulacella birmanica is the pest which developed resistance against ACH and DDT in the year 1963. Then different pests has developed resistance, and it was reported from different parts of the India. The cartworms, Spodoptera litura, its resistance against ACH was reported in the year 1965. Then the notorious pest, diamondback moth, Tutella gylostella, it has developed resistance against almost all the available insecticides. Even diamondback moth has developed resistance against the BT. It has been also reported in the year 2000. Diamondback moth resistant against BT karstaki, and it was reported in the year 2000. Even see the stored grain pest, red fur beetle, Tribolium castaneum. It has developed against DDT Malachion, and the resistance was reported in the year 1971. So the basic question is, somebody may be thinking that the pest has developed resistance, it is fine. What is the basic questions of what is the basic definition of the resistance? So there are many definitions are available in the resistance as far as resistance is concerned. But the most acceptable resistance to a layman We are layman, the resistance is nothing when a insecticide fails to give the satisfactory result or the expected result, then they may suspect that the pest has developed resistance. But as per the definition of the World Health Organization, they are telling that resistance is nothing but the genetic ability of an insect population to tolerate the dosage of insecticide that would have killed most susceptible individuals of the same species. This is the definition given by World Health Organization. Now see the order-wise the percentage sharing of the resistance. If we see the pie chart, easily seeing that we can easily see that the Diptera order and the Lepidoptera constitute more than 50% resistant species from the Diptera and the Lepidoptera order, and the Homoptera constitutes only 15% of the resistance cases. If we see the resistant species, agricultural pests, 63%, about 63% agricultural pests have developed resistance, and as far as the medical and veterinary pests have developed, 29% of the resistant species has been developed. Not only this, even see the pollinators, even see the predators, they have also developed resistance, but the resistance cases are reported very less or few number from different parts of the world. So there are the two types of resistance. We know that cross-resistant and multiple resistance. Let us clarify these two terms with some examples. It will be very very easy for us to remember. Cross-resistance, when a population of the housefly became resistant to DDT in the year 1950s. That is, the housefly was reported resistant to DDT in the year 1950s. Also exhibited resistance with no previous exposure record to pyrethroid insecticides used decades later. That is, DDT was introduced earlier, but pyrethroid was introduced far later, but still the housefly showed resistance to pyrethroid insecticides. So DDT and pyrethroids have the same mode of action. So they have developed cross resistance. That is, cross resistance can be defined a resistance phenomenon where a pest species or organism develop resistance against the insecticide or chemicals which has never been exposed to that particular chemicals. Now the multiple resistance. Multiple resistance, when the multiple resistance mechanism present or when a pest become resistant against the insecticide, against insecticides having different mode of action. There's some example, just see. Localized population of the Colorado potato beetle are notorious for multiple resistance to more than 50 insecticides with various mode of action. So these are two terms usually we use in toxicology, that cross resistance and multiple resistance. Now see the arthropod families with maximum cases of the resistance. It has been found that Noctuidae family developed the maximum. These are the some families where maximum cases of the resistance have been reported, like Noctuidae, Culicidae, and Aphididae, Plutilidae, and Tenthrididae, Tetranicidae. These are some families which has developed resistance. Maximum resistance has been reported from this family only. Now, an important data: top 10 countries by number of resistance cases. You see, the USA is the country where maximum number of the resistance have been reported. Resistant cases have been reported, followed by Pakistan, and then China, and then Australia, and India ranks 5th in the number of documented resistance cases. In India, more than 500 cases of resistance have been recognized or have been reported in different parts of India. Next, top 10 resistant species in the world. If we see the world scenario or the global scenario, we can easily see that two-spotted pygosmite, that is Tectronicus artici, it has developed resistance almost against 93 ingredients. See the diamond back moth, it has developed against 82 ingredients. It has developed varying degree of resistance against 82 chemicals. Green peach aphid, Myzus persicii, it has developed 74 the resistance against the 74 ingredients. Colorado potato beetle, housefly, silver leaf fly, that is Bemisia tabasi, cotton bollworm, Helicoverpa armigera, and Blatta germanica, cattle tick, Bupilus. Almost all these are some pests which have developed against the all available ingredients or chemicals for their management. See the regions what behind the development of the resistance in these 10 species. There are several regions, but some important regions may be that intensive selection pressure. This is the most important factor. Several research have proved that the major reason behind the development of the resistance is intensive selection pressure. Intensive selection pressure by way of frequent application of the insecticide, by use of prolonged use of the insecticide, having same mode of action, So we are putting a huge selection pressure on the organism, on the pest, so that they developed resistance within a very short period of time. Their polyphagous nature, exposed to many pesticides on many crops. So these are some reasons. Even the asexual reproduction or high reproduction rate, if you see the Titanicus artici, their high production rate, or Oligonicus coffee, their high reproduction rate, their availability throughout the year, these are some of the reasons, and the high dispersal rate. These are some of the reasons which are responsible for development of the resistance. Now, the basic question, the mechanism of the insecticide resistance. What are the mechanism of the insecticide resistance or insecticide tolerance, whatever? But we have to understand the mechanism behind the development of resistance. Otherwise, we will not be able to manage this problem very tactfully. So there are various mechanisms, but we will discuss some one by one. See, first development resistance mechanism is the altered behavior. So this is the basic phenomenon to any organism. Whenever you will put some selection pressure, they will try to avoid. Be it human being, be it insect, be it mite, where any organism try to avoid the adversity. The first level. This is the first level of the insecticide resistance mechanism. When the insect encounters an insecticides, they try to avoid the coming into contact with that insecticide. So this is their innate behavior. This is their innate behavior to avoid that particular surface where it is treated with the particular insecticides. So see some example. Empuasca flavescens, or it is called tea jacet, or it is also a pest of castor. Empuasca flavescens avoid exposure to direct sunlight and therefore prefers to stay on the underside of the tea leaves. This particular behavior cross protects the pest from the direct insecticide exposure in conventional tea plantation during spraying. So these particular pests usually stays underside of the leaves. So this behavior, they usually protect them from direct touch or from the direct contact with the insecticides on the leaves. I will see another example, Sartothis dorsalis, which resides inside the leaf bud during the development of the tea and underside of the leaf during adult stage, thereby avoiding direct exposure to the insecticides. So during their immature stages, they are staying inside the leaf bud, and when they are coming or becoming adult, they are staying inside the underside of the leaf during the adult stage, thereby avoiding the direct exposure to the insecticides. So these are their behavior, which by the way, this behavior giving them protection against the direct contact with the insecticides. So this is the one of the first level of resistance mechanism to the insect. Now, development of the barrier tissue. Once the insect comes in contact with the insecticide, so fine. First of all, they try to avoid the spaces or the places or the surfaces which is treated with the insecticide. Once they come in contact with that particular surface or particular insecticides, a reduced and delayed penetration through the cuticle will reduce the effect of the insecticide at the target site. A reduced and delayed penetration through the cuticle will reduce the effect of the insecticide at the target site. The mechanism may not prevent the insecticide from the eventually entering the insect. That is, this particular mechanism, that is the development of the barrier tissue, it will not prevent the insect from entering the insecticide. I mean, the insect, this particular mechanism will not prevent the insecticide from entering the insect body, but it can reduce the rate at which the insecticide reaches the target site. In female Helopeltis thivora, a higher level of body lipid has been found, which effectively reduces the penetration of insecticide to the target site. So higher body lipid content in the female Helopeltis thivora has been detected, which actually act as a barrier in entering the insecticides into their body. So this is one of another mechanism of development of the resistance. Now the most important mechanism is the enhanced detoxification. So now the insecticide has entered into the body of the insect. Now what will they do? They will enhance the detoxification, higher metabolism of the insecticide through higher metabolism of the insecticide. Before entering into the metabolism of the insecticide, let me show something, the basic structure of metabolism inside the insect body. See, when an insecticide come into the insect body, then they form the primary products. Now the insecticide, after entering into the insect body, undergoes the metabolism, and which is or detoxification through higher metabolism. This is consisted of two reactions, phase 1 reaction and phase 2 reaction. Every reaction is mediated by some enzyme. So some enzyme in the phase 1 reaction are the MFO or mixed function oxidases, esterases, be it acetylcholine esterase or carboxyl esterases. And in the phase 2 reaction, there is a conjugation reaction which is mediated by glutathione S transferase. It is also an important enzyme. Then the conjugated products is formed, and ultimately these conjugated products is excreted out of the insect body. Pest management failure occurs due to changes in the susceptibility level of the pest species to the applied insecticides. Pest management failure occurs why? Because the changes in the susceptibility levels of the pest species to the applied insecticides. Susceptibility level changes mainly due to the metabolic detoxification of the insecticide through higher level of activity of some insecticide detoxifying enzymes. Resistance due to the enhanced activity of detoxification enzyme have been reported in Helicoverpa, Scodophthora, Bemisia, Oligonicas, Tetranicas, Flutella. Everywhere the elevated activity of the detoxification enzyme have been reported. There are three major detoxifying enzymes. We all of you know that is a esterase enzyme, or your MFO or mixed function oxidase enzyme, that is cytochrome P450, and glutathione S transferase. These three enzymes are mainly responsible for detoxification of the xenobiotics or insecticides. So the higher activity has been found in the resistant species as compared to the susceptible species of Helicoverpa, Scodophthora, Bemisia, Oligonicas, Flutella. In most of the cases, these enzymes activity has been found, more activity has been found in the resistant species. So it can be said that these enzymes are responsible for development of the resistance. Then most insecticides which are water soluble or fat soluble are metabolized to less lipophilic or polar compounds, and ultimately they excreted out the body of the insect. Now the fourth important is the target site insensitivity. The last level of insecticide resistance mechanism is target site insensitivity. We know every insecticide has some target where they act. Suppose acetylcholine esterase are the target sites for your OP or organophosphate and carbamate compounds. Voltage gated sodium channel of the nerve membrane is the target of pyrethroid. Fipronil targets the GABA receptor. Imidacloprid targets the nicotinic acetylcholine receptor target site. Alteration at the target site to a less sensitive target for neurotoxic insecticide is an important resistance mechanism in the insects. So the studies have shown that when different resistance mechanisms are combined in the same individuals, a synergistic effect is created and ultimately resulting in a high level of resistance may arise. So these four mechanisms may present at the same time in the insect body, which ultimately gives a synergistic effect, and the ultimate level of the resistance actually increases. So resistance monitoring. So these are the mechanisms, four mechanisms we have seen. That is the altered behavior, then creation or barrier tissue, then metabolic detoxification, and then last one was the target site insensitivity. So these are some four major mechanisms by which insects become resistant. Now the resistance monitoring, how I will or all of we monitor the resistance. There are some important points which I will discuss now. That is resistance monitoring. First step is the sampling. We have to collect the sample of the pest or the organism from the field level. For that, we have to go to the field and we will sample. Suppose when a farmer sometimes demands that this particular insecticide is not working or it is not giving the satisfactory control, then our first work is to find out whether the pest has, we have to suspect that the pest might have developed the resistance. So the first work will be to collect the sample of that particular pest. So sampling is an important to constitute a representative sample of a location. Vary from pest to pest. Sampling procedure vary from pest to pest. For example, sampling of 1000 to 2000 eggs from widely separated fields to represent 4 to 5 kilometers of square kilometer of potential host would constitute a reasonable sample size. In case of tea, I have seen that actually 22 sample of Oligonicus coffee or sample of your Empuasca flavescens are collected around from 20 to 40 hectare land from different location within that particular area, and then they mix thereafter to, and that particular mixed population, that particular mixed individuals give the representative sample of a location. So first we have to collect the sample. Rearing, then we have to rear the particular sample. because standardization of the field collected population is required. Whenever we collect the population from the field, rearing techniques are specific for each species. Rearing is a 7 days week job. We have to rear the pest very carefully. Why we have to standardize the population? Because whenever we are collecting the pest from the field level, several variation will be there. That particular pest may be attacked by some predator, some parasitoid. So we have to rear the pest in the laboratory 1 or 2 generation. Then we will use it for bioassay for detecting the detecting or monitoring the resistance. So detection of the resistance. First we have sampled the population. Then we have standardized the population in the laboratory by rearing in an optimum environmental condition. Then we have to detect the resistance through different method. I will discuss. There are usually 4 detection method, but we will discuss here conventional bioassays and the biochemical assays. These are the most commonly used bioassays procedure for detection or monitoring of the resistance. Now, bioassays, before conducting the bioassays, some points should be kept in our mind, which is very important. First one is the stage of the insect. See, the toxic effect of an insecticide can be highly age and stage specific. For example, Lepidopteran adults and early instars are known to be more susceptible to insecticides as compared to the older stage larvae. In some experiments, we are seeing that we are using the third stage larva or third stage larva. In sometimes we are using some nymphal stage. Sometimes we are using some adult female stage. So that particular toxicity depends on the age and the stage specific of that particular insect. So we have to clearly mention that which particular stage of the insect or test organism has been used for the bioassay experiment during the publication of the paper. The choice of insecticide. See, Helicobacter pylori strains shown the more resistance to deltamethrin than to cypermethrin or fenvalerate. So Helicobacter pylori showed the more resistance to deltamethrin as compared to cypermethrin and fenvalerate. Hence, resistance monitoring with any specific insecticide molecule may not be representative of the entire class of compounds. If Helicobacter shows that more resistance to deltamethrin, and then we can't say that it is resistant to all the synthetic pyrethroid, because we have to be very specific to that particular insecticide, because that particular pest or population may be resistant to deltamethrin, but less resistant to other members of that synthetic pyrethroid, that cypermethrin or fenvalerate. So choice of the insecticides for it has to be clearly mentioned that this particular pest has been screened or has been assessed against these particular insecticides. Now the bioassay environment, it is necessary to maintain the optimum condition for the insect growth. If we give some environmental stress to the insect during our bioassay experiment, that may give different result. So bioassay environment is also very important. Now the diet. Unsuitable diet can vitiate or enhance the toxic response. It is important to ensure that the larvae get fresh diet in frequent intervals over the entire period of bioassay. Natural diets such as leaves or plants parts can be very different due to the age of the plant part, stage of the plant, and variety also. Even some paper, all of you will notice that they have mentioned that variety of the cultivar of the particular plant which was used for bioassay experiment, because the result of the bioassay may vary depending on the variety, depending on the stage of the plant also. So these particular points has to be mentioned very clearly, that the bioassay experiment was conducted against the stage of the insect, against which insecticide, against what was the optimum temperature, what was the optimum relative humidity, light and dark period during the bioassay experiment. Everything has to be mentioned in any bioassay experiment, and even the diet has to be mentioned. Now, health of the organism. Field collected larvae may harbor parasitoids and diseases which make them vulnerable to toxic effect of the insecticides. Unhygienic condition in the insectaries can also significantly impact the result of the bioassay. One of the recommended strategy adopted for the resistance monitoring is therefore to collect eggs of the target insect species directly from the field, rear them to the appropriate stage, and then conduct the bioassay. So these are some important points before conducting the bioassay experiment. Now, the sixth point, which is very important, I have seen some variation in the bioassay result. It depends on the operator skills also. If the insects are not handled delicately using proper forceps or fine brushes, it is likely that they will injure and the bioassay effects will definitely magnified. Careless technical work can ruin your bioassay experiment. So it is very, very important that operator skills are very important. Similarly, if the syringe is not properly rinsed between the each application of the different insecticides or even between different doses of the same insecticide, bioassay results may vary. So operator skills is very important. It is strongly recommended to start topical application from the low dose and then subsequently proceed to the higher dose. It is also true in case of your leaf method also. We should always start from the lower dose to the higher dose. Operator skill is very important. I have told you. So in case of the mite, when we transfer the mite from the infested leaves to the bioassay or to the insecticide treated leaves, we usually use a fine soft brush. After transfer from the infested leaf to the insecticide treated leaf, we have found that some mites may get injured during this process of the transfer. So we have to again re-observe. We have to re-observe whether any injured mite is there. If it is injured, then it should be replaced immediately. Then we should start bioassay experiment. Otherwise, the result of the bioassay experiment will definitely magnified, and it will not reflect the actual resistance or susceptibility status of the pest against a particular insecticides or acaricides. Now, the method of the bioassay. So these are some points which should be kept in mind before conducting any bioassay experiment. So bioassays, topical application method. We can see that topical application method, it is one of the most important or commonly used. In this case, the technical grade of the insecticide we usually use, and this particular technical insecticide is diluted using the acetone, and it is placed on the thoracic portion of the larva using a Hamilton repeating dispenser. Simply, the picture can be simply seen that how we use using the Hamilton repeating dispenser on the thoracic portion of the larva for bioassay. So we apply the topical portion of the particular larva. Now that IRSC method, different methods are there. We will describe shortly in brief. We will describe some methods. IRSC method number 5. This is recommended for green leafhopper and brown plant hopper. So nephrotic species and your Niloparvatalugens, we IRSC insecticide. IRSC usually recommended this procedure. See that particular pot, the base of the rice seedling has been sealed by using auger, and then that particular top, the particular plant is immersed in the insecticidal solution for 10 seconds. And then larva, then the neem for adult, whatever it is, GLH and BPH is released in each test tube within which the test plants are there. Then number of count and the observation on different days interval are taken. Then ultimately we go for bioassay, and then probability analysis is done, and then LC50 value or LC95 value or LC99 value we calculated using some software. Then IRSC method 8. This is recommended for whitefly. See, there are two plastic glass. The first one is, you can easily see that first one, inside the first one some water is there at the basal portion, and then second one that is a smaller which fits inside the first inside the bigger glass. You simply the leaf with some petiole should be kept inside the second glass or the smaller glass, and the mouth of that particular glass is closed with a muslin cover, and the whitefly leaves are inserted. Whitefly infested leaves or whitefly are inserted into the leaves, and then again mortality is observed at different days interval. So this is some method also. Or we can also use IRSC method test for whitefly, simply leaf dip method. Simply what we do, we simply dip the leaf in the insecticide solution, and that particular leaf is offered to whitefly for bioassay studies. One thing we have to keep in mind that the lid of the particular petri dish should be perforated, otherwise there will be accumulation of the water, and whitefly is very susceptible to the accumulation of the water, and they will ultimately die, and your bioassay result will be definitely misleading. Now the method IRSC method 20, it is a dyed incorporation method. Here some small trays are there, semi-synthetic dyes are poured inside it, and then along with the chemicals or whatever BT toxin, whatever it is inside that particular tray, and then larva is released in each tray, and their mortality is observed different days interval. Now this is the tip dip method. This method is observed. This method is practiced in case of the jacid. You can see that tip that it is a tea tip. Tea tip was collected from the tea bushes, and it was wrapped with a cotton, and then that particular cotton was water soaked. So these are the tips, and these are tips are air dried under the room temperature under a ceiling fan, simply in a perforated plastic container, and then this particular tip was immersed into the insecticide solution using a forceps. So it should be very careful during the immersion. We have to immerse the tip inside the insecticide solution for few seconds, and for the proper wetting of the leaves, and then particular leaves again air dried for the for drying of the surface liquid, and then we offer these tip into the for infestation of the jacid. That particular insecticide treated tea dip are kept on a plastic jar or plastic glass containing a perforated lid, and then jacids or imposca flavescens or whatever it is released inside the glass, and their mortality is observed at different days interval. Then a leaf dip method. Simply leaves are collected from the field, they are washed very carefully under the running tap water, and they are air dried in the laboratory. Then they are dipped into the insecticidal solution. Then these leaves are these leaves, what we do in the leaves are kept on a cotton pad, water soaked cotton pad, and the edge of the margin of the leaves are bordered with the cotton, water soaked cotton, and then 20 or 30 or 10 mite are released on the on oligonicoscopy is mainly released on the upper side of the leaves, and then their motility is observed under different days interval. So these are some leaf dip method. Previously was the tip dip method, and this one is the leaf dip method. There are several methods of bioassay. Now, after the compilation of the mortality data, we go for the abort correction. After abort correction, we go for probability analysis, and we get LC50 value or LC99 value, whatever it is. Now, after the next procedure is the calculation of the resistance factor. LC50 of field population of the mite and LC50 of the lab reared susceptible population. It is lab reared, it may be the most susceptible. Suppose I have collected whitefly from 5 different population from 5 different location. The most susceptible susceptible means the population which have the LC50 value. So all the we have collected the LC50 value. Then we will calculate the resistance factor based on this formula only. LC50 of the field population of any field population of the mite by LC50 of the most susceptible population of the mite. So resistance factor, or we sometimes also calculate the resistance coefficient by this formula LC95 of a population of mite by field recommended doses. So we can resistance coefficient, and we can say their scale are some there. If the value of the resistance coefficient is less than 1, then that is lack of resistance. If it is 1.1 to 2, low resistance, 2.1 to 5. These are resistance coefficient. Even for resistance factor, some scales are there for each insect, it may vary. So on the basis of that, we may find that which one is resistant or which one is susceptible. Suppose the LC50 value of particular location, suppose the LC50 value of the Bhubaneswar whitefly population is LC50 of 50 ppm, and the LC50 value of Kolkata population is 100 ppm. So we can say that the Kolkata population is 2 times more resistant as compared to the Bhubaneswar population. So this is the way we can say that this is 4 times resistant, this is a 5 times resistant as compared to the particular that susceptible population. That susceptible population may be a lab reared population of it, or it is not possible to get a lab reared susceptible population. Then we may compare based on the most susceptible population, most susceptible field population available to us. So these are the bio we calculate the resistance factor or resistance coefficient. Now the second most important is the enzyme assay. We always sometimes we use the enzyme assay for detection of the resistance. See some basic instruments which is required for conducting the enzyme assays. I have shown here that some basic instruments like your some electronic weighing balance, some magnetic stirrer, third the pH meter, micro pipette of different capacity, then micro tip, then one your ultracentrifuge, high speed ultracentrifuge, then your one spectrophotometer, or a microplate reader will be best for assessing enzymatic activity. Then one ice bending machine, then refrigerator and ice box. These are some basic instruments for conducting the enzyme assay. So now see the enzyme assay, preparation of the enzyme extract. Preparation of the enzyme extract, the first we have to extract the enzyme from the sample. Preparation of the homogenization buffer. First of all, we have to homogenize the insect sample or mite sample, whatever it is. For that, we have to homogenization buffer when enzyme homogeneous preparation. We have to prepare the homogenization buffer. Different homogenization buffer are used for homogenization of different insects. So we have to first, the work is to prepare the homogenization buffer, and using that homogenization buffer, the insect or mite sample will be homogenized for enzyme extract preparation. Estimation of the general storage activity. Now, general storage activity usually assessed using the alpha naphthyl acetate as substrate, following the standard protocol. For every insect pest, there is some standard protocol. We have to use that protocol. We have to verify it in the laboratory. We have to standardize in the laboratory. Then it can be used for estimation of the storage activity. Activity may be measured as a rate of the change in absorbance at optical density change in the spectrophotometer or your microploid reader at 450 nanometer over time. Electrophoretic analysis, sometimes we do electrophoretic analysis of the general storage like native page. Native page we usually do sometimes. Number and density of the bands may be analyzed. We may see a band for better understanding. See the first one. The first one, enzymatic or native page band. The first one, the susceptible population. See the band density. See the band density. Say very light, very light. This is susceptible population. See the Annamalai population or the Nilgiri population. See the Nilgiri population. The density of the band of that particular very black, or the density is far more. So that particular Nilgiri population is much more resistant as compared to the susceptible population. See the central term of the last one, central the band. So these are the some enzymatic or electrophoretic analysis of the acetylcholine enzyme for identification or for detection of the resistance. So we may also do the dense isometric analysis can be done for a better understanding of the resistance status of the particular location. Now, estimation of the glutathione transferase activity is also important enzyme. I told that this is very important in the phase reaction 2. Reaction phase 2. It is very important. Usually, we detect the activity of the glutathione transferase using 1-chloro-2,4-dinitrobenzene or CDNB as the substrate. Absorbance is usually recorded at 340 nanometer. Then the third, these are three major detoxifying enzymes. Then the third one is the P450, cytochrome P450 or MFO, mixed function oxidase activity. We usually estimate the activity of the MFO using p-nitroanisole as a substrate. Then the absorbance was recorded at 405 nanometer. Then the activity of the enzymes are calculated using some formulas only. Now the total protein quantification should be done of that particular sample. The total protein quantification can be done by 2 ways, either by Lowry method or by Bradford method. These are very easy method. We can easily conduct these experiments in our laboratory for easy detection of the resistance status. Now the most important is ultimately the management of the resistant pest. These are the most difficult job to any entomologist for management of the insecticide resistance management. Now, I like to start with a very simpler question. What is the best possible resistant management program? So many programs are available, use that. So to me, what is the best possible resistance management program? The answer is the simple, that is try to avoid the use of insecticides. That is the best answer for avoiding the insecticide resistance management. But someone will ask, sir, is it possible to avoid the use of the insecticides? If you can't give up the insecticides altogether, then the next best thing is to integrate chemical controls with effective cultural, biological, mechanical, physical, or genetic control to minimize the insecticides use. Anyway, that we call it, in short, we call it IPM. Any way by hook or crook, we have to reduce the selection pressure on that particular organism. Otherwise, it is inevitable that pest will develop the resistance today or tomorrow. The pest will develop resistance, it is true. So we have to reduce the selection pressure on the pest. Now the question is rotation of the insecticides having different mode of action. Sometimes it is recommended that use the insecticides having different mode of action. So we may follow the IRAC mode of action classification, and accordingly we can recommend to the farmers that use that particular insecticide and don't use the insecticides having similar mode of action every time or for a prolonged period. Rather, you use the insecticides having different mode of action. Mixture formulation may be used, but under careful consideration. Yes, I again stress that particular point that under careful consideration, mixture formulation may be used, but it should be used under the supervision of a particular expert. Otherwise, what will happen? See the phenomenon. Consider the residual period of the individual insecticides in the mixed formulation. In the mixed formulation, there are two different insecticides. Whenever we use in the field, then different particular insecticides or the member or individual insecticides have different waiting period, have different residual period. So we have to consider that before applying our mixed formulation insecticides. Each insecticide of a mixture should not have cross resistance to population of the resistant pest concerned. Suppose we are going to maintain, manage the resistant population, so we have to consider the cross resistance also, whether that particular individual insecticide in the mixture, whether they have that cross resistance in that particular resistant population. We have to ensure first, then we should recommend the mixed formulation. Since the components of a available ready mix formulation, have different mode of action and detoxification routes, the possibility of development of cross resistance may not arise. However, we can't stick to this point firmly because the deltamethrin resistant strain of Helicoverpa armigera had developed positive cross resistance to chlorpyrifos, even though mode of action of these two chemistries are entirely different. So Helicoverpa armigera had developed positive cross resistance against chlorpyrifos, even though the mode of action of these two chemicals, deltamethrin and chlorpyrifos, are different, but they have developed resistance against these two insecticides. So there is a report of the positive cross resistance. So we have to take care of this example also. Since the effect of the pesticide mixture is considered more toxic than their individual components, extra care should be taken to reduce the health hazards. Combination products should not always use as a matter of routine spray. It is need-based use should be ensured. Otherwise, there is no effective mechanism to restrict the use of the combination product or the mixture product. Every time they will use it, and it will ultimately increase their cost of cultivation, and it will ultimately increase the problem. Application of the insecticide must be timed correctly, targeting the most vulnerable life stage of the insect pest. This is also important. Management failures, we should also find that management failures is not only due to the development of the resistance. There are some, suppose a farmer comes to me and asks me that this particular insecticide is not working, then first of thing, we should not think that yes, it is development of the resistance. It is not like that. We have to think several other factors before concluding that it is due to the resistance development. We may see the timing of the application, we may the number of the application, the dosage, the correct application method, environmental condition may also affect the control efficacy. Rain soon after application may cause loss of insecticide. So there several factors need to be considered before saying yes, this particular pest has developed the resistance against the particular insecticides. This is all about pesticide resistance management. Thank you. This is all about pesticide resistance. So let us take oath that we have to manage the insecticide resistance management, because see, resistance management is inevitable. It's inevitable. It will happen. It is we will manage the resistance phenomenon. We have to reduce the selection pressure on the pest. By this way, we can definitely win the problem ultimately. Thank you everybody for.
Thank you, sir.
Thank you very much, sir. You have given very informative and very detailed presentation on how to manage the insecticide resistance developing in the insects. Sir, thank you very much, sir. On behalf of our Department of Entomology and Pathology, we are very grateful to you that you have agreed to present on this webinar, sir. Sir, we'll move forward with some doubt sessions. Some of our participants are having some queries and questions, so that we'll convey it to you. Okay, sir. Deepan, sir, can we move forward with the question? Deepan, sir, can we move forward with the question, sir?
Hello, sir. Good afternoon, sir. Am I audible, sir?
Yes. I am audible.
Yes, sir. Thank you, sir, for your nice deliberation, and we learned so many things from you. Thank you, sir. Very nicely you have explained all of the things, sir. Some of the queries are coming, sir. First thing is some question has came like these chlorpyrifos and some of the insecticides which were banned, though they were having highly efficacious, but still they are banned, sir. Madam is asking what should be recommended now, because generally this, for example, chlorpyrifos, it was having much more use, but now that is banned. So what should be recommended instead of that?
Our recommendation, several other new chemicals are there in the market. See, we have to consider the first generation insecticides when the DDT, chlorpyrifos, all the red level insecticides, they were very, they are effective definitely against the target pest, but they have also some impact on the environment. They have some impact on the pollinator. They have some impact on the earthworm. So, but the new generation insecticides, they have reduced impact on that particular beneficial insect. So, be a beneficial insect or some beneficial organism. So, we definitely have to shift to the new generation pesticides. Although the pest or the cost of the new generation pesticides sometimes more than the old generation pesticides, but we have to shift. Against chlorpyrifos, several other pesticides like flubendiamide, very good effective, spinosad, spinotoram, indoxacarb, several pesticides are there. We may go for that, but before that, we have to ensure whether that particular insecticide is recommended against a particular insect or pest or not. We have to first ensure that yes, it is recommended, then we may go for it. And before that, we have to again learn that whether that particular pest has any resistance status against the particular insecticides or not. Then we should go for recommendation. Is it okay?
Yes, sir. Just the thing is, Dr. Madhusmita Madam, she has asked for some of the list, sir. So not now, but after the session, if possible, you can share with us, sir, some of the recommended.
Yeah. No problem. I can say I can share you the recommended insecticide, recommended insect, and the dose and the waiting period, which is most important. Waiting period should be adhered to the. I know that most of the farmers don't know about the waiting period, so we have to be very careful after for recommendation. It is very difficult to recommend an insecticide because we should have knowledge about the waiting period, about the MRL, all those things before recommending, and the resistance status. We never usually don't consider the resistance status of the pest. In our region, I have seen that some farmers always some farmers say that, sir, imidacloprid is not working against the white flies, or what should I do? Yes, you have to have some type of information whether that particular location or particular population of that pest has developed resistance against a particular insect or not. So we should have that particular information and then go for the recommendation, not directly jump into the recommendation. Now, I know how many farmers are very aware about the ETL. What is the ETL of the particular pest? We say usually use the pesticide at ETL, but many farmers don't know what is the ETL status of the particular pest. So all these things should have recommending insecticide requires so much information. History of the application of the pesticide, history of the resistance status, all these things need to be ensured, and then definitely we go for recommendation.
Yes, sir. Thank you, sir.
I will share. Definitely, no problem. I will share, Dr. Nihal. You please share with madam.
Okay.
Thank you, sir.
If any participants has any queries,
sir, excuse me, sir. From our YouTube streaming also, some questions are there from the chat box. Sir, Dr. Mahfouz, he has asked some of the question, like what is the ideal and favorable weather for sucking pest and whitefly?
For
sucking pest and whiteflies also.
See. The question is the ideal or optimum environment.
Yes, sir.
See, what happening? I am following that nowadays that the particular whiteflies are a menacing pest in around the year, particularly. You see in tomato, but the population gets increased just before pre-kharif or just before rainy season. If you go to the in our place, you can see lots of whitefly on different pests, especially brinjal. It is a havoc infestation is there. So it is actually what happens nowadays. These polyphagous pests are occurring around the year. Round the year they are occurring.
Okay, sir. Sir, his second question was recommendation for insect resistance management. So you have very nicely told all those things. Sir, his third question is potential bioagent for whitefly at field. He is asking potential bioagent for whitefly. He is asking.
Potential bioagent for whitefly.
Yes, sir.
See, some pneumonia relay or some fungal formulation is there. But I am not sure about their efficacy. I am not sure about their efficacy. But if somebody knows it, may share it. But it is better for going some insecticidal application or some resistant varieties or some botanicals. All these things, for due to we have, because we usually suggest for IPM now, so we have to go different. So no, these are some pest, some formulation which is very effective against the whitefly.
Thank you, sir. Sir, from the chat box, we got another request. Dr. Vijay Kumar sir has asked, sir, can you share the PPT with us? He has asked for that in the chat box.
See, I can share with Dr. Nihal. Please,
Yes,
if somebody needs, see, as a
Sure, sir.
toxicologist,
I will distribute it to all the participants, sir.
yeah. The thing is that see, they can share the PPT, no problem. But the thing is that whatever it is there, not all the work is mine. So I have mentioned, I have
Yes, sir,
greatly
yes, sir.
acknowledged the source or the references. Please, whenever you do something, please acknowledge their source, their hard work. Always use this reference as a reference. Also mention the source from where you have got the information. It is an honest request.
Thank you very much, sir.
Thank
Any
you,
question,
sir.
please? Anybody?
Yes, sir. Any questions?
Sir, from the chat box, we have completed the things. From participants' side, we can start.
Okay.
Sir,
Any
first of all,
participants having any questions, you can ask us.
yes, sir.
Yes,
Sir, I have one question.
please ask.
Yes, please.
Sir, thank you, sir, for such nice informative section. I'm Dr. Sudhanshu Aranai. Sir, I have one doubt. What is the insecticide resistance status in case of natural enemy?
Yes. I have shown in a graph that even the predator, even the pollinator has also developed the resistance, but not so much work has not been done. Actually, not so much work has not been done on the resistance status on the beneficial organism particularly. What is their resistance? Not work. You will not. If you study the literature, not much work will be available, but some reports are definitely there. I have seen it in a graph. Only some few percentage of the resistance has been reported from predators and some pollinators. You may find it in the slide only. I will share the slide.
But sir, it will be helpful, no? Sir, meanwhile, if they will develop resistance towards insecticide, so it can be helpful for farmers' point of view also.
Yeah, definitely it will be great helpful. Otherwise, it is a recommendation of the pesticide. If you recommend a pesticide which is not effective at all, then it is of no use to the farmer. So
sir, is there any method to develop the resistance towards insecticide in case of natural enemy?
Yes, see, it is our duty to work on that particular, the resistance status on the natural enemy. I am going to work on that particular, but the literature, not so much literature is available on the resistance status of the natural enemy. You may find lots of paper on Helicoverpa, Spodoptera, Prutella, whitefly, some storage pest, but the paper on the resistance status on the natural enemies are very, very few. We have to start the work. Definitely, they are a living organism. They might have developed some resistance. We have to explore that.
Okay, sir. Thank you, sir.
Okay, thank you, Dr. Naik.
Sir, excuse me, sir. Sir, actually, am I audible, sir?
Yes.
Sir, again from YouTube, one request is coming, sir. They have requested, Satish sir, he has requested to send the list of new generation pesticides names, ETL levels, and waiting period. He has asked for that, sir.
Yes, I will share the name of the insecticide, against which pest it is recommended, at the dose, both the technical grade and your formulated product, and the waiting period. I will share, don't worry. Okay?
Okay, sir.
I will share with Dr. Nihal. I think he will share with all of you who requires it.
Yes, sir.
Okay.
Okay.
So Satish
Any
sir
other
can
queries?
contact Nihal sir and get that information. Thank you, sir.
Okay, I'll be sharing you to all your WhatsApp groups. All the PPTs and the documents will be shared to your WhatsApp groups.
Okay.
So I don't think that's all. No other queries are there, sir. Sir, thank you very much, sir. On behalf
Thank
of
you.
our
Thank you,
Department of
everyone.
Entomology and Plant Pathology, we are very grateful for you, sir, for
Thank you.
coming and having a talk with us.
Okay, take care, everybody. Under the present context of your
Okay.
COVID, take safety first and do research work. Okay, I use a healthy
Yes,
life
sir.
of everybody. Thank you, everybody.
Thank you, sir.
Thank you very much, sir.
Okay, thank you.
I would like to thank all the participants for attending this webinar. Thank you, one and all. Tomorrow, the third day of the webinar, will be starting from 11:30 a.m. The same link which you use for today's webinar can be used for tomorrow also. Tomorrow, 3 speakers will be presenting: Dr. Subrata Datta from BCKV, Dr. Amalendu Ghosh from IRI, and Dr. Krishna Kamakar from BCKV. So, hope to see you again tomorrow. Thank you all for joining the webinar. Thank you very much. Now I'm going to conclude the webinar.